Hello everyone. In this class, we'll try to study about the skeletal muscles. Skeletal muscle is made up of many long thin cells called muscle fibers. Each muscle fiber is multinucleated 1 to 40 millimeters long, cylindrical and having a diameter of 50 to 100 microns and surrounded by a cell membrane which is called as sarcolemma. Muscle fibers are made up of many fibrils called as myofibrils, and each myofibril is 1 to 2 micrometers in diameter. The cytoplasm in the muscle fiber is called sarcoplasm. Light microscopic appearance. The cross striations which are characteristic of skeletal muscles are due to the difference in the refractive indices of various parts of the muscle fiber. Therefore, the muscle fiber is seen to show alternate dark and light cross bands. The dark band is made up of highly refractile material which is called as A band, that is anisotropic band, which is having a length of 1.5 microns. In the center of each A band is found a slight less refractile region which is called as H band. M line is seen in the middle of H band due to the central bulge of A band. The alternate light band is made up of lower refractile material, therefore it looks lighter and is called as I band, that is isotropic band, which is having a length of 1 micrometer. In the center of each I band is found a narrow line, highly refractile material which looks dark called Z line. The contractile unit of the muscle is the substance between two adjacent Z line which is called as sarcomere and having a length of 2.5 microns. This is a diagram showing a sarcoplasm. The sarcomere is the area between two Z lines, and each sarcomere they are made up of myosin bands which are thick and actin filaments which are thin, and so they have been divided into dark band and light bands. Dark band is A band, whereas I band, the light band is I band, which is anisotropic. Electron microscopic view. The myofibrils are made up of two sets of protein filaments, thick and thin filaments. Thick filament. It is made up of myosin, which is responsible for the formation of A band. Myosin molecule is made up of 6 polypeptide, 2 heavy chains and 4 light chain. Myosin treated with proteolytic enzyme, it reveals 2 fragments. Short compact head of meromycin, which is called as HMM, it has two important sites, that is actin binding site where myosin comes in contact with actin, and there is the ATPase site that hydrolyzes the ATP. Long chain of light meromycin, it has only structural function and it acts as a backbone for HMM. This is the structure of the myosin, where it has got the heavy chains and the light chains, 4 light chains, and this is divided into head part and the tail part, and head contains the actin binding site, and it also has a site for ATPase activity. Thin filament. It is made up of actin, tropomyosin and troponin. They arrange themselves to form 2 chains of globular units that form double helix, which is responsible for I band. Each thin filament contains 300 to 400 actin molecules and 40 to 60 tropomyosin molecule actin. It occurs in 2 forms. The actin occurs in 2 forms: G-actin, which each molecule bind with 1 molecule of ATP firmly, and F-actin is formed by the polymerization of the G-actin. Tropomyosin, it covers the binding site of actin where myosin head comes in contact with actin. That is, it prevents the interaction between actin and myosin. Troponin, they are small globular proteins made up of 3 subunits. Troponin T, which binds the other troponin components to tropomyosin, whereas Troponin I inhibits the interaction of myosin with actin, and troponin C contains binding site for calcium that initiates muscle contraction. Coming to the sarcotubular system, it is a highly specialized system of the internal conduction of depolarization within the muscle fiber. It is made up of T system, that is transverse tubular system, and a longitudinal sarcoplasmic reticulum, that is LSR. T system, they are inwardly directed extensions of the sarcolemma into the muscle fibers at the junction between A and I bands. Its function is the rapid transmission of action potential from cell membrane to all the fibrils in the muscle. The T-tubule protein is modified voltage-sensitive calcium channel known as dihydropyrimidine receptor, so named because it binds the class of drugs called as dihydropyrimidines. The main role of DHP receptor, however, is not to conduct calcium, rather to act as a voltage sensor. Longitudinal sarcoplasmic reticulum is longitudinally lies on the either side of the tubular system are found the sacs of the dilated LSR. These sacs are called as terminal cistern, which are rich in glycogen and calcium. Calcium channels in the sarcotubular reticulum are not voltage gated and allow the release of calcium into the cell. These are called ryanodine receptors. Transverse tubule with the two terminal cisterns are called as triad. There are two triad per sarcomere, and action potential on the T-tubule cause the DHP receptors to induce conformational change to open the ryanodine receptor channel. Calcium is thus released from the lateral sac of sarcoplasmic reticulum. This calcium binds with troponin, removes the blocking action of tropomyosin. So this is diagram showing the sarcoplasmic reticular system. This is the transverse tubules and these are longitudinal tubules. The transverse tubules helps in the conduction of action potential towards the longitudinal reticulum, where the action potential causes the activation of dihydropyrimidine receptors, which mechanically activate ryanodine receptors, and ultimately it leads to the release of calcium into the sarcoplasm, and this calcium is going to bind to the troponin, which is going to initiate the muscle contraction. The steps of excitation contraction coupling. The process by which depolarization of the muscle fiber initiates contraction is excitation contraction coupling. Sequence of events in excitation contraction coupling are the steps involved in the muscle contraction. So in the beginning, there will be stimulation of the motor neuron that causes the release of acetylcholine into synaptic cleft, causing generation of end plate potential. Then there will be depolarization of the muscle membrane by increasing its permeability to sodium. Then there will be generation of action potential in the muscle fiber. Then there will be inward spread of action potential along the T system. Then spread of depolarization to the terminal system with the release of calcium in the myofibrils. Then there will be increase in the concentration of calcium in intracellular fluid by as much as 2000 times, which is going to lead to the muscle contraction. Role of calcium. Calcium binds with troponin C, causing tropomyosin to move laterally. This exposes the binding sites for myosin heads on the actin. It activates the prosthetic group of myosin filament, which acts as an enzyme, which is ATPase, myosin ATPase. It also helps in the steps in muscular relaxation. A few milliseconds after the action potential is over, sarcoplasmic reticulum begins to reaccumulate calcium. The calcium ions are actively pumped by calcium magnesium ATPase into longitudinal portion of the reticulum to terminal cistern for storage. Molecular basis of muscular contraction, which is also called as sliding filament theory or ratchet theory. The process by which the shortening of the contractile elements in the muscle is brought by is the sliding of actin filament over the thick myosin filaments. The sliding of filaments is brought by the formation of the cross bridges between the head of myosin and actin molecules. Events which are occurring during the formation of the cross bridges. In resting muscle, no cross bridges are formed since troponin I is lightly bound to actin and tropomyosin covers the actin sites where myosin bind to actin. Therefore, tropomyosin complex constitutes a relaxing protein which inhibits the interaction between actin and myosin. When calcium released from the terminal system by the action potential, the calcium binds to troponin C and it causes the binding of troponin I to actin is weakened. Because of this, troponin I to actin is weakened, and this permits tropomyosin to move laterally, uncovering the binding sites for myosin heads on the actin filaments. Then there will be hydrolysis of ATP by ATPase actively in the myosin heads, which produces energy. 7 myosin binding sites on actin filaments are uncovered for each molecule of troponin that binds a calcium. The heads of myosin molecules linked to the actin produce movements of myosin on the actin by shielding. Then mechanism of sliding of thin filaments or thick filaments. The sliding during muscle contraction is produced by breaking and reforming of cross bridges between actin and myosin, repeating the process in a serial fashion for at least 5 to 6 times. Then the width of A bands is constant, whereas the Z moves closer together when the muscle contracts. It leads to shortening of the I bands. This diagram showing the position of the troponin I and the binding site of the myosin over the actin. When the calcium binds to troponin, the binding site of myosin head for actin is exposed, and the myosin head is going to bind to the actin. And this diagram shows the power stroke where it is showing the ratchet theory or sliding theory. Okay, so when the myosin head binds on the actin filaments, then because of the ATPase activity, the power stroke occurs which pulls the actin filaments towards the M line and which causes the shortening of the muscle fiber. Motor unit. Motor unit is the functional unit of muscle contraction in intact body. It consists of single motor neuron cell body, its axon fibers and muscle fibers innervated by it. The cell bodies of the motor neurons supplying the skeletal muscle fibers lie in the ventral horn of the spinal cord or the motor cranial nuclei. In simple words, each single motor neuron and all the muscle fibers it supplies constitutes motor unit. So this is the diagram showing the motor unit. Motor unit is nothing but it is the number of muscle fibers which are supplied by single spinal motor nerve. Properties of motor unit. The number of muscle fibers in a motor unit varies inversely with the precision of movements performed by that part. Each motor unit innervates only one type of muscle fibers. In other words, in a single motor unit, the muscle fibers supplied by it are of same type. There are two types, that is red type, that is also called as type 1, and white muscle fibers that are also called as type 2 muscle fibers. All the efferent fibers passing to skeletal muscles are excitatory. There are no efferent nerves which on stimulation produce relaxation of the muscle. That is, there are no inhibitory efferents to skeletal muscles as compared to cardiac and smooth muscles, where efferent supply is both excitatory and inhibitory. Depending on the type of muscle fibers they innervate, motor units are of two types, slow and fast motor units. Coming to the applied aspects, when motor nerve to a skeletal muscle is cut, it causes diffuse atrophy of the muscle. That is, it causes shrinkage of the muscle fibers, which finally gets replaced by fibrous tissue. Or there can be complete paralysis of the muscle. And there can be appearance of fine irregular contraction of individual fibers called fibrillation. There can also be de-innervation hypersensitivity. These effects are the classical consequences of lower motor neuron lesion. That is the injury to the spinal or cranial motor neurons which directly innervate the muscles. Muscular dystrophy. It is a syndrome characterized by progressive muscle weakness due to the mutation in the dystrophin gene, which results in congenital defect in dystrophin glycoprotein complex. This is a large protein complex that connects the thin filaments to the transmembrane protein in the sarcolemma and thus provides support and strength to the muscle fibers. Rigor mortis. When muscle fibers are completely depleted of ATP and phosphocreatine, they develop a state of extremely rigidity called rigor. When this occurs after death, the condition is called as rigor mortis. In rigor, almost all of the myosin heads attach to actin, but in abnormal, fixed and resistant way. Myotonia, a condition characterized by difficulty and slowness in relaxing muscle after voluntary effort. Various forms of myotonia defined clinically are due to the abnormalities or abnormal genes on 7, 17 and 19 chromosomes, which results in abnormalities of sodium or chloride channels. Types of muscle fibers. Muscle fibers can be divided into two types, that is fast muscles or slow muscles. The fast muscles are also white muscles and slow muscles are they are red muscles. Fast muscles are pale looking whereas slow muscles are more red. This is so because the slow muscles have greater capillary network. In human beings, most muscles contain variable mixture of fast and slow muscle fibers. The postural muscles are largely slow muscles. White muscles. White muscles constitutes 3/4 of the total muscle mass and have a greater speed of contraction. They have a resting blood flow of 3 ml per minute per 100 gram, which may increase to 60 to 70 ml per minute per 100 gram in maximal exercise. These muscles are prone to oxygen debt. Red muscles. They comprise 25% of the total muscle mass and are concerned with maintenance of posture. Their activities of the study and prolonged type which requires relatively low oxygen usage. Their resting blood flow is 20 to 30 ml per minute per 100 gram due to the low basal myogenic tone. They have vascular bed 3 times the size of white muscle and high flow capacity, approximately 150 ml per minute per 100 gram. Because of greater surface area of the capillary bed and their lower oxygen requirement, these muscles are unlikely to be exposed to oxygen debt. So these are some of the differences between the white or red muscle fibers and red muscle fibers. The white muscle fibers are large in diameter with high glycogen capacity and ATPase activity. They are called as white muscle fibers because they are pale. Whereas red muscle fibers are moderate in diameter with moderate glycogen capacity and low ATPase activity. They are called red muscles because they are darker than the other muscle fibers. White muscle fibers are fast muscle fibers because they are innervated by large, fast conducting motor neurons. Whereas red muscles, they are innervated by small, slow conducting motor neurons. White muscle fibers get fatigued easily, whereas red muscles They are resistant to fatigue and are most used muscles. Whereas the white muscles are specialized for fine and rapid skill movements and have short twitch duration. Whereas the red muscles, they respond very slowly and have a long latency period, and they are adapted for sustained slow and posture maintaining contractions. For example, the long muscles of limb and muscles of back. White muscle fibers particularly suited to perform high intensity work that can be sustained for only short period of time, whereas red muscles, they're suited to perform low intensity work over a longer period of time, such as for athletes and swimmers. So this is in brief about the skeletal muscle. Thank you.